Welcome, everyone, to our webinar today. We're very excited to talk to you about our strategic alliance for bioconjugation and ADC drug substance and drug product manufacturing services, really showing off the power of two, with two companies that are powerhouses in this area and the ease of one single integrated project. So today, we'll talk a little bit about the offer from MilliporeSigma and Simtra Biopharma Solutions. The agenda we have for today will focus on three major deliverables. So first, we'll talk a little bit about Millipore ADC and bioconjugation CDMO services. Then we will focus on Simtra Solutions fill-finish CDMO capabilities, led to us today by Lupe Reyes, and then delivering what matters the most, all the workflows and examples that make it an efficient process for our clients. So for the first topic, we'll really focus in on what are the capabilities at MilliporeSigma in St. Louis. So here we provide a single organization which really has a global network in the ADC value chain. So on the screen in front of you, you see the different sites that contribute to the value chain, and we'll go around the map around the world here and kind of talk about what each one of the sites do. First, let's focus in on Schaffhausen, Switzerland. This is our facility which really focuses in on functionalized PEGs. So that is important as an intermediate in drug linkers, whether you're putting in a linear spacer in your drug linker or you're actually putting on a branched PEG to be able to change the lipophilicity and shielding of your payload linker itself. Those can be very critical and valuable intermediates that go into the synthesis of the drug linker. Then if we look over into the United States, our Madison, Verona sites are our HPAPI manufacturing facilities. That's really nice for us in St. Louis because that means we have a direct line into getting the payload linkers and being able to influence the quality and all the specifications that we need to be able to do the bioconjugation in St. Louis. In St. Louis, Missouri, as we've kind of referred to, that's the BioCenter Excellence for Conjugation in MilliporeSigma. I won't spend a lot of time here because I'm going to spend a lot of time on it as we move through the slides. We'll speak a little bit more about the capabilities in St. Louis itself. And then finally, we do have, of course, our Rockville site, which is contract testing services, which also has the capabilities of doing ADC testing. All right, a little bit about our team. So I am the director. I have the pleasure and honor of leading the process and analytical development team in St. Louis, where, of course, we focus on bioconjugation as well as a class of compounds called bioorganics. So we are very similar. I hope you hear the same story from every development team. It is our job to deliver robust and comprehensive methods for both manufacturing processes as well as analytical testing into our GMP organization for custom projects. We support and continue development of these custom projects all the way from GMP1. So the team is put together to be able to develop and introduce it into our GMP manufacturing facility. We also have teams that specialize in late stage 1 validation, getting ready for commercial production. And whether it's a good or a bad thing, our team stays engaged throughout the entire life cycle of that process. So we really spend our expertise being able to understand to depth all of the chemistry and bio unit operations that go with handling ADCs. We do have 10 specialized teams in St. Louis, so the development team is quite large, and we have the ability to kind of focus on different chemistries. So we have folks that really kind of work on that cysteine-lysine conjugation chemistry. We also have a large group of people who understand enzymatic conjugations, whether that's the glycan remodeling platform, TG, sortase, all of the enzymes that are used now or site-specific conjugations, as well as some of the novel or non-cytotoxic, non-potent conjugates. So that's important because as we see new and novel payloads being introduced into these delivery mechanisms, it's those payloads that change the phys-chem properties of those conjugates, and understanding what that is how we now can develop analytics and different unit operations that handle those new phys-chem properties. In addition to that, our team has a long history in what we call bioorganics chemistry. So this can be small molecule chemistries that really have some kind of water solubility, either purification step or solubility in water as their final composition. Those are polyamino acids, polysaccharides, polynucleotides, and lipids. And that's important because that was in our history before we actually started doing bioconjugation. So this team understands very clearly the interactions between small molecules and large molecules and how to be able to manipulate all the physical chem properties to be able to control those substances. We are also very lucky if you look at the next item, which is called differentiating technologies. So not only do we have experience in all of the bioconjugation techniques that are out there, we do have state-of-the-art analytical methods for characterization, and we'll show some of that in the workflows later. In St. Louis, we do 95% of our own testing to be able to release as well as characterize an ADC. And that can be very important when we're trying to do troubleshooting also, so that we have all that methodology available to us. Bioanalytical methods here, I mean things like ICE, CGE, LCMS work, SPR, all of the types of testings that are really looking at some of the structural activities that come with the protein. And then I'm also very lucky in that our direct staff, we hire people that usually come out of large pharma for our management organization. They understand CMC. They've all been in the development pipeline at large pharma companies before. And that means they can help your team as well as our team really kind of guide that maze about how do you get a drug through the development pipeline and then into a both an IND and commercial organization. A little bit more about our experience. So within the development team, and this is going to, of course, telescope then into our GMP production, over 150 different constructs and over 1,000 batches in the development space alone. Our scale is quite large. So we do everything down from a MIG when we're doing DOEs and different OFAD experiments, all the way up to 1,500 grams. This has become especially important as we've moved into that novel conjugate space because toxicology batches are getting larger and larger because we're not on the cytotoxic platform all the time. So being able to deliver that GLP tox still within the development space is something we very much are specialized at. We also have significant experience in chromatography. Over half of our constructs have chromatography in place. Sometimes it's around protein remodeling, sometimes it's around differentiation of species as we do isolations of those constructs. But the team is very specialized in both screening as well as transferring that information over into our GMP manufacturing organization. Here we see the broad conjugation chemistry history that we have. So we've done conjugation both stochastic as well as full reductive. We still do some lysine chemistry, mainly around chelators, but that is something that is still in place. But I would say the majority of the work over the last 5 to 7 years really has been focused on understanding site-specific chemistries, bispecific analytics, as well as looking at the different enzymatic ways of being able to do conjugations. So here, engineered cysteine, so the whole Thiomab series, anything that's enzyme assisted, whether it's TG, sortase, non-natural amino acids, of course, which kind of leads into the middle click chemistry. Engineer tags and understanding of how to show that we are in fact only hitting those tags, and then glycan remodeling platform. We do quite a bit around that. Payloads, cytotoxic, as we've already talked about, but we also work very heavily in immunotoxins, oligonucleotides, dyes, antibiotics, chelators. Bioconjugation has really changed from just cytotoxic constructs into looking at any kind of payload that you specifically want to deliver to a tissue in a body. All of those things are in play now for bioconjugation techniques. Additionally, we have been on the single-use reactor platform since 2017 in PEDX ourselves. So we were very lucky as part of the Millipore family. We were able to influence our colleagues to be able to make an ADC reactor that has not only just everything that we need for bioconjugation, but also our full PAT platform that we're quite skilled here in St. Louis. Of that 150 different constructs we've worked on, over 80 of those have been transferred into our manufacturing organization. Of course, that means 80 INDs, but what that means to my team is 80 INDs times the amount of patients that have been impacted by all of those studies. So we're very proud to say that, we're having significant impact on the health of all that patient population. And then manufacturing, 15 plus years of expertise, over 350. I know it says 300 there, but I just got the count yesterday, 350 batches, and about 40 of those have been commercial batches with an ongoing capacity expansion. A little bit about analytical capabilities too. So we'll talk a little bit about core analytics. So really the full package comes for release here. So you can see anything that has to do with standard technologies around UPLC, HPLC, GC, and compendial testing. Every one of our standard offers also comes from our bioanalytical team, which we have a specialty team that works on ICGE, SPR. We have in-house LCMS to be able to look at both protein as well as large molecule. MFI and SECMOL's COE is actually part of the St. Louis site. In collaboration with that, we also have some additional resources that come from our hub. So things that are partnered with what we would call our ARD team are listed there. So things that go into your characterization package. So intact LCMS, peptide mapping, glycan, higher order structure, anything around free thiol. All of those happen right here in St. Louis within our footprint. And then additionally, we don't let you go when we just get GMP1 out the door. Many times you will need things like tox dose verification studies, NU stability, infusion sets studies, all of those types of things. Our team also helps with those quite a bit. And then finally, before I hand it over, I did want to talk a little bit about the recent investment in MilliporeSigma in St. Louis, over $91 million being invested in that center of excellence. This is upgrades to benefit PAD, so our particular labs to be able to expand our resources and our capabilities, as well as expansion for QC, R&D, manufacturing, and our logistics department. And for those of you, worked on that plant floor, logistics can be just as important as sometimes sweet time. So we want to make sure we have a full build out of new labs, buffer prep, cold storage, and all of our warehouse spaces. This increases our manufacturing capacity, and we really see that as a response to the growing demand that we see for ADC and bioconjugates. We all do see this too as a stepping stone in our overall journey. So we won't be stopping here. We'll continue to grow so that we can meet the demand that you have for us. And with that, I'll turn it over to Lupe to take us through the Simtra capabilities.
Thank you, Lisa. Great learning more, obviously, about the Merck Millipore capabilities. So now I'd like to do just a brief introduction of myself. So my name is Lupe Rojas. I'm a business development executive within the Simtra Biopharma Solutions team. I'm responsible for obviously supporting existing clients or potential new clients as they look to assess and evaluate if they'd like to transfer a program over to one of our two respective sites. So it's a pleasure to be here with everyone. So just a little bit about who Simtra Biopharma Solutions is. We are a global CDMO with really a singular focus on sterile injectable manufacturing. And we're focused on leveraging our long-standing history and track record of producing commercial products, bringing them from clinical to commercial, and leveraging that expertise in support of our pharma and biotech companies, and offering what we call a full service continuum for drug product manufacturing, from preclinical formulation all the way through to manufacturing and commercial manufacturing as well. So a little bit about how we do that. So we are not new to this space. When we partner with our clients, we bring over 65 years of sterile manufacturing expertise, supported by over 2,500 employees across our two respective manufacturing sites. Between those two sites, we produce over 70 commercial programs that are distributed over 120 countries in support of 60 plus pharma and biotech clients. What I like to say is that we feel like we were on the ground floor with ADCs. We have over 10 years of experience producing clinical and commercial ADC compounds on behalf of our clients. And what I'm really, I'll say, most excited about is the fact that we produce over 150 million units distributed across the globe. And the key piece that I love about what Simtra is doing now is the investments in both capabilities and capacities that over the next 3 years total over $500 million in investments. So a little bit overview of the actual platforms that we support within our two respective sites. So as you can see here, going left to right, we support prefilled syringes, nested cartridges, liquid and lyo vials. And the types of products that we manufacture are cytotoxic, high potent compounds, including bioconjugations and ADCs. And as the industry has evolved and moved into what we call large molecules, obviously the majority of what we get nowadays is biologics, but we also support small molecules. And we actually do produce some diluents for our clients in support of their liquid or lyo vials from a reconstitution standpoint out of our Bloomington site. So how do we do that? We actually support our clients really on two key sites. And what we're looking to do as we continue to grow and develop is offering matching capabilities in both the US and the EU. So the two sites that we have one main site in Bloomington, Indiana, that has two essentially facilities. One called the main campus, which is has been in existence for over 25 years. And then the other one is our park campus. That site was newly acquired about middle of last year. And that site will now support ADCs in the US, once it becomes available middle of next year. In addition to that, we have our site in Halle, Germany, which also supports ADCs, high potent products. That's been their specialty for quite some time, but we've also expanded that and now are offering what we typically say is the more standard biologics and also pre-filled syringes moving forward. So what do we look to offer our clients? So we've been hearing more about having a global reach, a focus of our clients saying they like to see and have options where they can actually produce products in the markets they sell them to. So we can now do that with our two respective sites in Bloomington and in the US. And that offers them scale and flexibility and expanded capacity as their products go from clinical all the way to commercial. If you look at what we mentioned in terms of our overall expansion, so today we have 11 filling suites within our two respective facilities. By late 27, early 2028, that will grow to 18. So you can see quite a big expansion underway, over 50% growth in terms of the overall number of manufacturing lines that we have available. And like I mentioned before, our focus is offering both development all the way to fill finish services at both sites. And then we have the option in particular at our Park site for what we call build to suite custom manufacturing. So what does that mean? That means that if there's a specific, we'll say, need or capability where you require some sort of custom offering, we have the space to grow with our clients with that Park acquisition that we made a little over a year ago. So now digging in a bit about how we do and how we perform with ADCs. So really when you think about ADCs, a lot of these programs come in preclinical. So if you study the market a bit, you know that there's a significant amount of ADCs in development in that preclinical stage, growing into clinical and then going potentially into commercial. So we have the ability to support the full suite from a drug product standpoint. And I'm proud to say that we've actually transferred in over 60 ADC programs to our site in Halle, Germany, which includes 6 commercially approved ADCs that are distributed globally. So how do we support our clients from a drug product standpoint? We really focused on, like I mentioned, having the ability to go from clinical to commercial. So we have 2 clinical lines that are really designed. When you think about ADCs, the actual bulk is very, very expensive. A lot of times in the early stages, there's minimal bulk. So our clinical lines are really designed to support those type of programs by offering the ability to go to as small as 2,000 vials, but then also minimize the waste in terms of the actual setup and startup, because we do know that each vial is critically important as people start to go from that clinical early phase 1 and beyond. And then if the programs are successful, we have the ability with the 3 commercial lines to scale them up to the right level that's needed. And the benefit of that is that you're doing it at one site. So that provides customers the value of less transfer, less resources required, and also the background knowledge of the product itself and the understanding of the 2 teams already working together. That brings tremendous value in our mind to our clients. And that's why we've been able to transfer over 60 ADCs and counting as we move forward. So a little bit about the partnership. So we talked about, obviously, MilliporeSigma, the extensive capabilities they have. And then you look at us at Simtra. So the way we view this, Lisa mentioned this, we're bringing really 2 powerhouses together in the focus of doing really one key thing here, which is to streamline the overall process and accelerate the delivery of ADCs. The actual supply chain for these products is very complex and many times can go from one continent to the next. So if there's an ability to be able to bring our 2, I'll say, expertise together to streamline and accelerate that process, we believe that's going to bring tremendous value to our clients and really speed, I'll say, the go-to-market efforts to bring value to those clients as we move forward. So how do we look to do that? Like I mentioned before, we're looking to have a streamlined solution. So if you look at this from left to right, you can see from the beginning, both Simtra and MilliporeSigma are working together in terms of understanding the client's needs. So from project initiation and contracting, project kickoff, and then even the quality, I'll say the technical agreements and the assessments that take place there, those are all done in conjunction with each other in close collaboration. So we understand what's happening on the MilliporeSigma side, and then they also understand the downstream effects that would have for us. So we know that in alignment with our clients, Then we go into the drug substance development, which obviously the MilliporeSigma team would handle. And then we've also focused on getting our two teams together to understand how do we quickly receive and understand when product's coming in, so that when we get it, it's set, ready to go, and it's in a position for us to be able to take that and move it quickly into our filling suites. What Simtra can also do is we can also do the drug product formulation development if there's any needs or requirements there. We have our Lyle Center of Excellence in Bloomington, but we've also transferred that expertise over to our Hollis site. So if there's any Lyle development that can bring value to you in terms of making you a more robust product, or also reducing Lyle cycle, we can do that for you as part of the overall process. And then last but not least, obviously the drug product manufacturing for that product as well. So talking about what the partnership brings to our clients, so we see multiple benefits that can take place here in reducing complexity, shortening those timelines, and then creating efficiencies between the alignments that our two parties have together. So when I mentioned before a simplified supply chain, we're really focused on the two teams came together and identified where is there overlap, where can we cut and streamline processes, that making sure that we're both fully aligned. So that's what we've already done as part of that process. The other key piece is that you have, I'll say, designated PM teams that understand each other's processes. So we can, again, communicate quickly, align with the teams. If there's changes upstream or downstream, we can affect those immediately. We can also do conditional releases as we enter the fill finish. And then that also helps us synchronize when the bulk is essentially ready. We can be ready on our side to receive it and then quickly move it into fill finish manufacturing. I noted before our processes. So again, we're talking about alignment in conjunction on proposals. We do our proposal, they do their proposal, but we move in conjunction. We have a seamless transfer. And then if there's any sort of formulation work that's needed on either side, we can do that. And then the other key piece that I do want to just highlight for the team is that from a logistical standpoint, there's full transparency across all three, I'll say, parties. And that does bring value in streamlining processes. And then also as we receive product, we're able to understand exactly what's needed and move quickly in that process. So here's just a quick example of what we highlighted in terms of the early initial phases. When we first do an assessment, you can see here on the left, all three parties get together. We've essentially assembled a simple three-way CDA that allows us to communicate. Our expectation is that the three parties would get together and have joint technical calls to fully understand the full scope of what's required. And then our two respective organizations will develop our proposals, our statement of works, our MSAs, our QA agreements, all in conjunction. And you can see at the bottom, the program management teams are working together to obviously initiate POs as needed, get dual kickoff meetings going, a combined kickoff meeting, and then also project initiation. So again, the focus is on how do we streamline, how do we align our efforts to reduce and compress that overall timeline for our clients. Now I'm going to turn it over to Lisa so she can actually walk through the process itself and how the two parties are going to work together to, again, enable our clients.
So thanks, Lupe. Our goal right now is to kind of take you through some of the workflows. And my intent is when we get done with these next couple of slides, you would almost feel like you've worked through a project and could really walk away with a very concrete example of what this partnership can bring to you today. So for this slide, you're going to see the combination and how we move the materials back and forth. So in yellow are going to be the things that would be executed at the MilliporeSigma site in St. Louis, and in green would be the Simtra organization. And then the purple bars are going to show where we were doing things together or where maybe there's a material transfer or a GMP start. So if we start at the left side, of course, we've already developed at this point, if we're going into GMP manufacturing, the process at MilliporeSigma. So we have good understanding of the product and how to control it, all the analytical methods and all the unit operations that go with that production. So we would bring our GMP material in, we would do our DS testing, and then of course that material would get a C of A and released by the MilliporeSigma site. That material would then get transferred over to the Simtra organization. They would get the DS receipt. They would do their raw material ID testing, their incoming raw material testing. And then, of course, that would go over into their GMP DP fill manufacturing. Along that same time, the BDS is still at MilliporeSigma up in stability. So we'll be doing the stability testing, gathering more product information over time, And all of that DS stability report matrices come out of the quality system in the St. Louis site. And then if we switch back over at the same time, and that's the power of this, right? Things are happening concurrently and at the same time. That GMP DP manufacturing is ongoing. And after that is done, this is where we have a little bit of a split. So if you look in the middle of the screen there where it says DP testing with both of our colors, we'll give you some more information about what that entails, more details about that. But what we're really talking about there is some DP testing that is much more specific to the DP testing at Simtra, and the things that are more product specific that have already been developed at MilliporeSigma for the ease of having just one testing site is happening at the MilliporeSigma site. MilliporeSigma will present a CFT that will go over to Simtra for final DP certificate and release testing. And along that same path, as the DP comes back, that will go to St. Louis. We will be doing the stability testing as well as presenting all that information back through a stability matrix report. So if we look at some of the advantages to that, we see that the DSDP package really is all under one roof. So it's under one virtual roof, yes, right? But it's transparent to you because we are handling it for you. You don't have to worry about informing Simtra or informing Millipore about what's happening in your process. Additionally, all that fill and finish capacity has been reserved because they have a pre-read. They know exactly through that program management system that Lupe talked about what's happening with the program and where it stands. And then finally, testing is happening in parallel. So yes, while some samples are moving back and forth between the two sites, we are keeping the expertise where it needs to be so that we can facilitate the testing as quickly as possible. All right, so the next slide, I want to kind of take you through how the program actually walks through from the development all the way through to manufacturing. So we're going to build a slide here together. And it's going to be the same kind of color coding for you. So yellow is going to be MilliporeSigma, green is going to be Simtra, and we're going to watch the movement back and forth between the two companies. So when we start a project at Millipore, the first thing we're going to do is develop some analytics and start doing some tech transfer and process optimization. So at that point, we're talking, Simtra's on those calls. They know when the things are happening and coming to us. And as soon as we get through a tech transfer, as soon as we have API to send them, that BDS is going to Simtra for doing either lyo development or for doing liquid fill, looking at that too. So that analytical method transfer is happening at that point. So anything that's on our platform methods that Simtra is going to need is being automatically transferred, as well as small scale first look kind of samples. Then as we move forward, they will start looking at non-GMP feasibility fills, and we will be starting to look at things like optimization, scalability, facility fit into the BDS GMP organization. And then again, as we make those demonstration batches, whether it's going to be a tox fill, whether it's just going to be an engineering feasibility fill, we're talking with Lupe's organization to say, how much do you need? When do you need it? What format, what bag, what container do you need it in? Then they will be working on that DP technology transfer to their GMP organization. And then finally, when we get to GMP scale, of course, that technology transfer is going to happen where we're sending material from both development for those feasibilities as well as from our GMP manufacturing facilities. So here we're going to, think positive. We're at PPQ here, but this also stands in place for early phase supplies. And so that material would, of course, get transferred over. Then we would have that DP done in HALA. And that material would then be comparability tested, characterization, as well as all of the comparability studies that are done at Simtra. Then we'll also just go forward into when we get to that PAI inspection, all of that knowledge has been shared back and forth, and all of the information that you would need to be able to fill out your BLA and be able to do your inspection is available. So that's the workflows, the two workflows, whether we're talking about the back and forth between all of the testing or we're talking about the development space. What I wanted to do now is spend a little bit of time to give you some more background on that box that I highlighted for you, where testing is happening at Simtra as well as at Millipore. And the real goal here is I'm going to start on the right-hand part of your slide in the purple box. The real goal here is no duplication, right? So we want to be efficient as possible. We want to share the information, but we don't want to be duplicating things. And if for the folks that have worked in this space for a long time, as soon as you have the same method, even if it's looking at different parts of the process and you have any kind of version control, either with analytical methods or you have different results, that can be a real nightmare to try to understand. So we're really trying to minimize the method transfer and make sure that we're providing a very uniform and consistent way of being able to release your drug product. So here, Millipore will execute the DS testing, and we will own the stability protocols. We will develop them, of course, in collaboration with Simtra. That conversation happens all the time. Simtra will execute DP compendial testing and any DP specific testing. All right, now I'll switch us over to the left-hand side of the chart. I know there's a lot of information here, and we're going to pay attention to colors more than we're going to pay attention diving down into individual things. And what you'll see here is when we talk about DP-specific testing, things that are phys-chem properties, things like Carl Fisher, appearance, pH, physical particles, those will remain at Simtra, right? Those will remain where the DP expertise and that DP side has it. We can do those things at MilliporeSigma, but we feel like it's a better fit and more efficient to do it right after the DP is made. And then you will move into things that we would call more ADC product specific testing, ICE, CGE, SEC, looking at free drug distribution and DAR distribution. Those things would still happen at MilliporeSigma. Part of the rationale for that is that method already exists at MilliporeSigma because we did the development of the construct as we went through there. So we're not going to ship it over to Simtra. We're going to hold it at MilliporeSigma and just consolidate the testing in one place. In the middle column, you will see the BDS testing. So because that's BDS, that would rely primarily at Millipore. Then we go to DP release. And you will see, again, the product-specific testing happening at Millipore. And then in the green boxes, you see things that are much more LIO or much more DP-specific testing. I will highlight in the identity box there, you see the one little shift there. That is because CDMO sites are responsible for showing that what they receive is different from other things on their site. So we will share, of course, what our site ID test is with Simtra, but it is possible that that would not differentiate away from the other things that they have on site. So there will be a due diligence activity to make sure that our ID methods are specific for each of the sites. And then when we get to DP stability testing, you can see the matrix is pretty much the same there. As we get DP lots back that are going up on stability, MilliporeSigma would do the ADC specific testing and Simtra would do the DP specific testing. We believe that this is really the most efficient way to be able to move your projects forward as quickly as possible and make sure that we have really good control of the product. With that, I hope that we've been able to kind of highlight how this integrated offer has already been worked out for you, that it's very easy to be able to take a product through this workflow. And I will say thank you and turn it over to Lupe for our final comments.
Thanks, Lisa. Yeah, I'd just like to thank obviously all of those that joined today's call. Obviously, we're excited about this alliance. We do believe that it will bring tremendous value to our respective clients and look forward to meeting with some of you in the future.